Hey guys, welcome back to a new video by Biology with Sangshuan. So today we are going to discuss the Cambridge IGCSE Biology, Paper 4 Theory Extended, the 0610 Paper 4 Variant 3. So this is the October-November 2024 series. So again, as a disclaimer for this video is that the answers in this paper will be in point form, but during your exam, please make sure that you write in paragraphs. And again, the answers that also I have is suggestive only, and some of your answers can be also correct. So please make sure that you cross-check with the relevant resources to make sure that your answer is also correct and also validated right too. Okay, so let's start with the first question, shall we? So question 1, figure 1.1 shows the part of the human digestive system. Okay, so using label lines and the letters shown, identify on figure 1.1 the organ that produces bile and label it as A. So, of course, remember, this is something that the students will also confuse. Producing is always the liver. The one that stores it is the gallbladder. But of course, they're not asking you to write. They're asking you to label them. So label in this form, okay? A pointing to the liver. Please do not draw any arrowheads. You are supposed to draw a straight line only. And B is pointing to the gallbladder, all right? So just as simple as that. You don't have to complicate yourself. And describe physical digestion. When they ask you to describe physical digestion, is they asking you for the definition of physical digestion. So which is actually the breakdown food into smaller pieces without any chemical changes inside. Okay. Part 3. State the name of two places where physical digestion occurs in the body. So first one is the mouth. Second is the stomach. We can also have the duodenum also doing physical digestion because it does not involve any chemical changes yet. Part B. Bile is involved in fat digestion. So complete the sentence about fat digestion. So you can see that almost all papers have filled in the blanks. So you must expect this kind of question to come out in your exam. So bile is released into the small intestine in the digestive system. Bile increases the surface area of fats and oils. So this is called as emulsification. This increases the surface area and increases the rate of chemical digestion using the enzyme lipase because they are talking about fat. So you must know that the enzyme that's involved here is lipase. So state the name of the products of fat digestion. So what are the two things that after fat has been broken down with lipase? So you can expect fatty acids and glycerol. Part C. Explain the role of bile in the breakdown of protein by trypsin. So how does bile allow breakdown of protein by trypsin effective? So first thing, bile is alkaline. So once it's alkaline, whatever is coming from the stomach, which we call as the chyme, is acidic. So the bile helps to neutralize the chyme from the stomach. Okay, and what happens is that it provides a suitable pH or an optimum pH for trypsin to function properly. Question 2. Aphids are arthropods. Aphids insert their mouthpieces into the phloem and in the stem of a plant to feed. And aphid is shown in figure 2.1. So I'm pretty sure that if you've done any past questions, you are familiar that aphids are the one that actually puts their mouthpiece into the phloem and take up sucrose and amino acids. So they're asking you, identify two features visible in 2.1 that you can classify an aphid as an insect. So first, you can see that there's three pairs of legs. Three pairs of legs you can see, and one pair of an antenna. That is always the most certain answer that you can get. So always when they ask you how to classify whether it's an insect, whether it's an this particular arthropod, you always consider the pair of legs that are involved and also whether it has antenna or not, because I think that's the most visible things that you can see easily from the diagram. Okay, figure 2.2 is a diagram of a cross-section of a dicotyledonous stem. So what you can see here is that this is actually a common stem that you can see, and state the letter in figure 2.2 that identifies the structure form which aphids will feed. So of course it will be the phloem. Phloem will be found at the top. So phloem on top, xylem on the bottom. Part 3. State the names of two nutrients transported in the phloem. So like I mentioned before, amino acids, sucrose. That's it. That's the two main nutrients that you must know that is transported inside that phloem. So part B, scientists investigated the effect of temperature on the rate of translocation. So calculate the percentage change in the rate of translocation between 15 degrees and 30 degrees. Give your answers in 3 significant figures and your space for working. Alright, so again, another percentage change question. So percentage is to see between the value initial and the final temperature, the value, did it go up or did it go down? So looking from here, it become from 7.3 to 17.1, meaning that this is showing an increase or a positive percentage change. So in your answer, you're supposed to have positive value and not negative value. You can see in my previous video in variant 1 of paper 4 of the October-November series, you can see that percentage change becomes negative because the value drops. Alright, so how do you solve? Using the formula final minus initial divided by initial times 100. You're going to get 134.24 by asking for 3 significant figures. So you put as 134. Alright, so that's your answer. So part C, figure 2.3 shows a potato plant growing in the spring and then in the summer. So they show you that this is the growing shoot, this is the tuber, and what happens, this is the tuber in the summer, and these are the growing plants. Okay, then the leaves that you can see here. So explain how translocation changes between spring and summer in the potato plant shown in figure 2.3. So use the terms source and sink in your answer, meaning the examiner wants to see these two words in your answer. So what happens during springtime? In springtime, you can expect that the growing shoot is the sink because there's no leaf, they can't undergo photosynthesis. And the one that is supplying all the nutrients is the tuber. So the tubers become the source. It gives you all the nutrients from the soil that is replenished by nature. And what happens is that they usually provide glucose. So glucose, the main thing, is helping for the growth of the plant, or you can say for respiration. But during the summer, you can see that the leaves are now able to photosynthesize. When they're able to photosynthesize, they can produce their own glucose. So as a result, the leaves becomes the source and the tubers becomes the sink, because this happens during winter where all of things switch again, and what happens is that all the nutrients that have been stored in the tubers will be used for growth or to make sure to maintain the plant properly. So as a result, this is the answer that I mentioned just now, that during the summer, the leaves are able to photosynthesize and then they can produce glucose. So part 2, new tubers form on the potato plants in the summer and they are genetically identical. So they've given you a clue that this is genetically identical. So you have to identify the type of reproduction that's occurring here. As a result, it's an asexual reproduction. Question 3. HIV can be transmitted from mother to infant. So describe how the risk of a mother transmitting HIV to their infant can be reduced. So first, the mother can have an anti-HIV viral medication, and of course reduce the chances of breastfeeding, because remember, if she has a baby or whether it's inside the placenta, the virus can actually transmit by crossing the placenta, and the baby can also get HIV too. Part B, part 1. So scientists investigate the effects of breastfeeding on the chances of getting an infection. So they studied 2 groups of 100 infants. One group was breastfed and one group was not breastfed. So you're using the table in table 3.1 describes the effects of breastfeeding on the chance of getting an infection. They are asking for you to describe. So just use whatever you can see from the values. So you can see that the number of infections that were seen, okay, the effect of breastfeeding is that there is reduced chances of gastroenteritis and also bronchitis infection. But you can see that it didn't change much for influenza and conjunctivitis. But although they are slightly higher, but the changes isn't that drastic. It isn't too drastic at all. Okay. Part 2. State the name of the white blood cell that's involved in the immune response. If you state white blood cell, lymphocytes, phagocytes, it's acceptable. Okay. And part 3, describe how infants can gain passive immunity. So passive immunity is by antibodies given from another individual. So you can see that involve antibody acquired from another individual. Breastfeeding is a way to pass the antibody to the infant. It needs regular antibodies so that the baby can have temporary immunity. And then later on, the crossing the placenta also can supply antibodies to the fetus. Part 4, describe how immunity gained by vaccination differs from passive immunity. So first, you state that vaccination is artificial active immunity. So inside the vaccine, there is weakened pathogens and the body is exposed to it so that the immune system can actually be ready to respond. So as a result, memory cells are produced so that there will be a long-lasting immunity. Question 4. Human muscle cells use anaerobic respiration during vigorous exercise. A part 1 state the word equation for anaerobic respiration. So they're asking you for the word equation. So please do not give the chemical equation. So as a result, in humans, there's only one product, no CO2. It's only glucose to lactic acid. That's it. Okay. So part 2, compare the process of anaerobic, to aerobic in human muscle cells. So you use anaerobic to compare, so use this as a standing point. So anaerobic, they do not use oxygen. They release lesser energy. In animals, they will produce lactic acid, and in plants, they produce alcohol, CO2, and H2O. That's it. All right, we have a graph question now. So an athlete runs a race which lasts 60 seconds. Figure 4.1 shows the heart rate and breathing rate of an athlete during the race as he recovers after the race. So you can see that while he's having a race, there's high heart rate, high breathing rate. And the moment he reaches about, let's say, around 125 seconds, it starts to drop. It starts to drop drastically, all right? So that's something that we can see from our eyes now. So the rate of breathing increases during the race is response to the change detected by the body. So again, where can you identify the stimulus and state the organs that detects this? So the stimulus is the increased concentration of the carbon dioxide, and the organ that responds to it is the brain. So now, part 2. Using the information figure 4.1, calculate the rate of decrease in heart rate from 125 seconds to 260 seconds as the athlete recovers. So this is much simpler. They're asking you for beats per minute. So you just find the beats per minute between the time here, okay, between here, and divide by the time, because they have already shown you that the unit here is beats per minute per second. So as a result, you're going to get 0.53. All right, so just put 0.53 or 0.5. It's okay, no worries. So part 3, explain the results shown in figure 4.1 from 60 seconds until 420 seconds. So what you can see at 60 seconds is that the heart rate, and the breathing rate will peak. You can see both of them. It peaks at 60 seconds. And after 60 seconds, aerobic respiration will occur. And what happens is that there will be lesser muscle contraction due to the oxygen debt, lesser energy is required, decreased need of oxygen, and heart rate will eventually return back to normal. So you're just going to explain what happens between this period of time till it begins to rest. You can see that everything goes back to normal the moment things starting to stop, and then things are starting to slow down a little bit, then everything goes down. So just talk about the process of returning back to normal state. Question 5. So we see another genetics question. So feather color in some breeds of chicken is example of codominance in birds. So in this example, a chicken with white feathers and a chicken with black feathers can breed to produce offspring that have white feathers and black feathers. The allele for black feathers is FB, and the allele for white feathers is FW. So they have given you all the clues here that these are the chickens that with the specific, all right? So state the types of variation that's caused by genes only. So caused by genes only mainly is discontinuous. Like your blood group, red blood, ABO blood group, this is all by your discontinuous variation and mainly by genes. So state all the possible genotypes of chicken 2 and chicken 7 in figure 5.1. So in chicken 2, they've given you something, a secret, okay, which we do not know. So what can we expect for chicken 2? Okay, remember this is also it should be a male, but we do not know what is it. But what you can see is that they have given you both black and white and the one that shows black. So what will be the possible genotype? You don't really have to go and do a Punnett square, but you can see that it either has FB or FB or FB or FW, because when you cross with this parent, you are going to produce all 4 of them. Okay, so these are the chances that they can get. So you don't really have to do a Punnett square here. And maybe for chicken 2 and chicken 7, now for chicken 7, when you cross with both, actually they are both considered to be heterozygous, but when you cross it, you are definitely going to get 3 different types. So FW, FW, FB, FW, FB, and FB. So this is the only one that you should do a Punnett square, because they ask you to list all the possible genotypes that you can find. So I'm pretty sure that you guys know how to do the Punnett square, so this shouldn't be a much problem, but it will be well suggested for you to do the Punnett square to solve this. Okay, so complete the genetic diagram to show the results between chicken 5 and chicken 6. So first, chicken 5 and chicken 6, they ask you for the phenotype. So get the phenotype from this. They're giving you the key and just copy inside here. And the genotype is always the genetic makeup. So it's always FB, FW. And gametes, you just separate. Remember, because gametes, you're going to separate 2 of them. Okay? One gives one gives another. It splits inside the gametes. And chicken 6 will also do the same. Okay, they will split gametes, okay, and do 2. So you do the Punnett square, and as a result, you should be getting 4 different things. But there will be a duplicate, so you can just stick with FB, FW, and FB and FB. That is enough. And the expected offspring phenotype percentage, 50% black and white, 50% black. So again, if you have any questions on this, please do make sure that you comment in the comment section below. I will actually help you out on this if you do not know how to construct the Punnett square. Alright, they ask you to state one example of codominance in humans. Like I mentioned before, ABO blood group is the most accurate that you can see here. Alright, question 6. So some blood clotting disorders can be inherited. So describe what is meant by the term inherited. So again, a definition question, and you must be able to have word by word. The important phrases has to be in there so you can get your full marks. So transmission of genetic information from generation to generation is sufficient for this question. Okay. Part 2. Describe the process of blood clotting. So when you have a bleeding wound, so what's going to happen? You have platelets begin to clot, because remember, platelets is the main fragment of the cell to do blood clotting. And what happens that there will be fibrinogen to fibrin. Okay. So what happens that fibrin becomes insoluble, and then it forms a mesh there so that it can trap the red blood cell. Okay. And part B, you can see that genetic modification has been used to produce human protein. So one protein made in the way is by this factor, okay, factor 2A, which is used to treat some blood clotting disorders. So what can you see as a result? Okay, human cells being extracted and it's being cut with the restriction enzyme, plasmid will be cut with the same enzyme, and what happens as the result? There'll be a cutting of the DNA and then joining of them, inserting the bacteria, and then it's grown in the fermenter. So state the names of enzyme A and enzyme B. So like I mentioned before, enzyme A is where you cut. So you use the same enzyme to cut both sides, and this will be the restriction enzyme. So for enzyme B, you are joining the two, both the human and the plasmid together. You are going to use a DNA ligase. They will help you to form the sticky ends to basically glue them up together so that you can form a recombinant plasmid. So part 2, explain why is it important that enzyme A is used to cut both the human DNA and the plasmid. First is because that you can form sticky ends. Second is that both of the DNA will be able to form complementary shapes so they can actually bind together accurately. Okay. So part 3, what happens after these have been stick? So the DNA ligase does its job, the restriction enzyme is cut the respective, and they have to glue them together. So what does it form now? That will be called as the recombinant plasmid. So part 4, explain why the contents of the fermenter in figure 6.1 are stirred constantly. So again, when you are blending a juice, or you're mixing the egg with the pepper, salt, everything inside, so you are mixing together. So what's the point of all of this? Is to make sure that there will be evenly distribution of the salt and pepper throughout the entire content. So same thing for here. All of it inside, you have fermenters, you have the nutrients, you have the gas, air, and of course the organism itself, and the necessary things that in order for it to survive, you mix them together so that all of the organism is actually able to have a portion of these important nutrients. Okay, so this is really straightforward. And bacteria can be used to make complex molecules. So why? Okay, what are the reasons that bacteria are useful in this case? Because it has a plasmid, no ethical concerns. These are the tools that I just stated. There are actually more, so feel free to state some of your answers in the comment section below. I can totally help you to check on this also. And part 6, okay, so describe 2 ways animal cells differ from bacterial cells. So they're asking you the difference between a eukaryote and a prokaryote. So it's a simple, animals have a nucleus, they have no cell wall. I think that is pretty much sufficient. If you mention no plasmid, also can, right? Okay, so crop plants such as soya can also be genetically modified. To discuss the advantage and disadvantage of genetic modification of crop plants. So again, these kind of questions, right, you definitely will know the advantages, but the struggling part is the disadvantage. But again, we'll go to the advantages first. So first, anything with genetic modification, you can increase a lot of things. You can increase the yield of the crop, increase the nutritional content, lesser use of the fertilizers, and what happens is that you can, it is also resistant to herbicides, so that you don't have all of these kinds of, this kind of side stuff, okay, all these kind of chemicals that's coming to your plants. You're keeping it natural. But for disadvantages is that having a genetically modified seed is not cheap. It's costly to some farmers. And of course, there's also ethical and religious concerns. Some ethical groups may find GMO seeds as a little bit, you are altering what nature is giving it to you, okay? And also, when you have genetically modified crops, they have no genetic variability. There's no sexual reproduction occurring. It's always asexual because you cannot mix with the wild crops, as you do not know what kind of adverse effects that's gonna bring from this kind of crops. Alright, so yes, that's all for this paper. If you have any questions, again, please do feel free to comment in the comment section below. I will of course help you to solve them as fast as I can, of course. So alright, so thank you so much for watching. All the best again, and see you in the next video. Bye bye.